All right, again, so we're going to start into the DSci presentation now. So lab.bio, accessible BioML tools, presented by Niklas from LabDAO. So please take your seat, and we'll get started here.
Thank you. All right, welcome everyone. I'm Niklas, founder of LabDAO and Convexity Labs, the company behind LabDAO. And I'm going to be talking about a use case on top of distributed AI, lab.bio. So the story started late 2020. I'm a recovering academic, and AlphaFold gets released. Who knows what AlphaFold is? Show of hands. OK. AlphaFold is a model that lets you design proteins or any other sort of biotherapeutic compound for that matter. And the question sort of came to my mind, why can't we design therapeutic candidates online? Like, what's in the way? And on the one hand, what's in the way is that a lot of the models that we can use to design therapeutics, these machine learning models, they're not accessible, they're hard to run, they're hard to operate. And then on the other hand, it's really hard to raise the capital to actually move a discovery that was made through one of these models along the clinical development pathway into an actual drug. So I think biopharma is such an exciting area to benefit from both progress in Web3 and AI, because on the one hand, progress in machine learning is making a lot of new therapeutic candidates way more readily accessible. And on the other hand, progress in IP tracking is allowing us to form capital and communities around discoveries way more easier than ever before. And that's why we built lab.bio. So we make tools for designing biology accessible, and at the same time, we make the discoveries themselves ownable. So we've been basically building something like a Hugging Face for biology, where we've onboarded multiple tools for protein binder design, protein folding, and other really biology and therapeutic discovery relevant models. And today I'm going to be telling you a bit of a vignette about protein binder design, so one particular use case for therapeutic discovery that we're real passionate about. And we learned a couple things that I think can be helpful for people that are being distributed AI platforms and that want to create a great user experience for their users, and I'm going to share some of the learnings that we've made. So the first one is, when you're using these BioML tools, there are a ton of settings. It's not just a word prompt, and a ton of output data as well. So when you build a platform, it needs to be a platform that can handle a lot of data. You cannot just host the models. That's why we built a user interface that lets you actually get a lot of granular control and run jobs in batches, actually highly parallelized, but also B, gives you a file explorer where all your data is pushed on IPFS. You can browse it, you can use it in your next model run, and you can share it with others as well. The second thing with biomachine learning is that actually only 1 in 100 designs work. So this is not like Midjourney, where you get a good picture after a couple iterations, or ChatGPT, where you have a response right away. You actually need to run the model a couple more times, which is really useful. Which is really useful if you think about decentralized compute, because you can dispatch a job onto multiple community nodes. They don't need to talk to each other, and they come back with the results. So just to illustrate this a bit, when you want to design a drug, let's say the screen protein here is a disease-related target, and you want to design a binder. Most of the time when you run one of these generative models, they will give you this thing here on the right side, which is a poor design. And you can see that the protein that we designed, which is in red here, it has very, very poor binding. There's this massive gap. Right? You don't need to be a biochemist to see that this is not necessarily doing its job. While on the right side, on the other side, you have a strong design where you see a lot of interaction. In order to find one of these, you need to run this model thousands of times to actually draw from that distribution. So we needed to build UI elements that let the user filter the experimental outputs and visualize the results. And this is just from one example that we ran, where you can see 2 different metrics that we get, both for the stability and for the binding affinity of a binder. And everything out from the other side of the red dot is actually a useful design. You can see just how many proteins that you're generating are not desirable, and you really need to run this for thousands and thousands of times to get something interesting. And this is where we use Bacalhau, a distributed computing framework that also supports GPUs to run models and give us the data that we need. So how does it actually look like? We generated about 3,600 binders. We selected 50 binders that were really useful, and then we went into the laboratory and we actually tested those. And we have about 5 to 6 successful binders. So you have a 10% success rate in the laboratory to actually design biology that is meaningful and that can serve as a starting point for a therapeutic discovery. But you have about 100x of just GPU costs and running a model, the same model every time, to find a binder that's even worth testing in the laboratory. The last thing that we learned, and I think that's very characteristic for the area which we're in, is that when you actually have one of these binders, there's a ton of financial value in what you discovered, but it's not yet there. It's not realized because you actually need to finance all the further experiments to take that one discovery into an actual therapeutic, right? It's got a multi-year process. So how do you help people that play around with these tools? How do you help them form the capital around their project, build the community they need in order to move their project forward? And that's where we work with records. It's an ERC 1155 that basically attaches to every job that you're running and captures both the rights to what you've done plus sufficient metadata to fully reproduce what you've done. So, for example, the metadata contains all the input that went into the model, the parameters with which you ran the model, as well as the output data. All of this is happening on the platform in a way that you own your work. So every time you run the model, you actually mint a token into your wallet. You create a wallet when you sign up. You collect all your data into your wallet, and you can easily pay for your jobs with fiat while you're doing all of this. Now, if you really like a project, and even if that project is not yours, you can discover this on the platform and you can sponsor it. You can basically tip someone for the work that they've done and enable them to move forward. This here is a short video where we just show how to use the platform in a short walkthrough. So here, we want to run a protein binder design experiment. We're selecting a protein that we want to start designing a binder against, setting some parameters, and then we can actually see it running on the distributed computing platform. So this is running on Bacalhau. The model is running relatively quickly, and then the results are becoming visible. We can browse them and eventually, visualize them in our wallet as well. So here we have a couple of designs. We can explore them on the platform and compare them, see their metrics, and eventually sort of move on into the next experiment. What we're working on right now is to actually integrate with physical laboratory services. So when you actually have something that you really like, you can raise additional funding and you can test it in the lab. And with that, I want to thank you for your time and happy to take any questions. Thank you. Yeah. Yeah, I hear you well. Yeah, I have my own point of view here, in which I think that a lot of these distributed protocols, they're really useful for inference, not necessarily for training, and at least that's what we're using them for. So when you're training a model, especially when you're training a large model, you have a lot of intra-GPU bandwidth communication that needs to happen because the model checkpoints get constantly updated. And that can be quite a problem when you're in a fully distributed setup because the latency is immense. You're not going to have a very effective training run unless all your GPUs are co-located. Now, in this particular instance, we're running inference. So the GPU, once it's running, is not dependent on any other internet traffic or exchange with other GPUs while the system is running. So we can shoot off 100 jobs in parallel. Every node can just compute on their own and publish the results without sharing of information. So I think I'm pretty bearish on distributed training. I'm way more optimistic on distributed serving and inference of models, which is what LivePeer and some of the other projects are doing. OK, thank you, everyone.